Hello, everybody. Good afternoon. Thank you for coming to this second event that we have here. And I'd first like to thank Dean Sharon Wood for letting us use this beautiful auditorium for this lecture series, and to thank Dr. Jim Truchard for providing the funds to allow this whole thing to happen, and to really initiating with his intense curiosity new ideas that might fertilize new discoveries in this field of neurodegeneration. So it gives me great pleasure to introduce a former colleague and friend, Tony Wyss-Coray. So Tony got his master's and Ph.D. in Switzerland, which is his home country, at the University of Bern. Then he came to the United States and did a postdoctoral fellowship at the Scripps Research Institute with Leonard Muche. And we recruited Leonard to UCSF, I guess it was 2011 or 12. Wait, no, 92. 2011 was different. Sorry, I need to listen to this lecture. 96, okay. So, and Tony was in the lab at the time, and he came with Leonard, who became head of the Gladstone Institute for Neurologic Disease. And Tony became a staff scientist in that institute and stayed there, and then got a position at Stanford University, where he rose to the ranks from assistant professor to full professor over several years, and was based at the VA. Now he has dual laboratories both on the main medical school, or the main university campus, as well as the VA, and has done extremely well in the field of neurodegeneration. His master's and PhD were in immunology, and so he brings an extremely strong background in immunology to the neurosciences. So he is a true card-carrying neuroimmunologist who doesn't study the standard neuroimmune diseases that we think about, the demyelinating diseases like multiple sclerosis, but really brings that basic science expertise into the field of neurodegeneration. So in addition to looking at risk factors, immunologic dysfunction as a risk or pathogenic factor in neurodegeneration, he's also interested in what seems like a very obvious but probably not as well studied question: is why are so many neurodegenerative diseases have as their major risk factor age? So what is it about aging that actually creates a risk for these disorders? So both immune dysfunction and aging have been the themes of his work, and I think he will talk to you about that today. And he's been using the latest tools of single-cell sequencing and what have you. His work has been recognized internationally, and he's gotten many, many awards, which I won't list. Some are listed in your brochure. And I look forward to your talk. Thank you so much, Tony, for coming.
Thank you, Bob. Thank you all. Thank you, Jim. Thank you for having me here. It's really exciting to be in this beautiful auditorium and to talk to you about the work we are doing. And as Bob already said, we're interested in neurodegeneration. And just to remind you briefly, this lecture is in honor of Oskar Fischer, who did actually the first real study on patients with presenile dementia, or what we call today Alzheimer's disease. In a way, Alzheimer's did a single case study, described one person, whereas Oskar Fischer actually had a whole cohort of almost 24 brains from patients who had this disease, and described these key features that we know today, these protein deposits that form, we call them plaques, and these threads of proteins that are called tangles. And he described them at the same time, but then didn't really get the credit of naming that disease. And that's a whole other history, but I just wanted to give that credit up front. So when you look at this picture and you're young, you don't really know why I'm showing it, but if you're a little bit older, you might have experienced problems finding your car in a busy parking lot like that. Anybody can share that problem? So if I ask the same question to my students, again, they don't know what I'm talking about, but as we get older, our brain has more trouble forming what we call spatial maps and retrieving these maps. And interestingly, animals have similar problems. Dogs, for example, monkeys, mice, and we can actually study mice to get specifically at this aspect of spatial cognition where your brain makes a map of the surrounding and can retrieve that map. And with age, we lose this function. It's one of the first or early signs in patients with Alzheimer's disease. So what my lab is trying to understand is how does this brain that can make these maps so quickly, and you never have to think when you're young where you parked your car, you just walk to your car, how does this brain become susceptible to age-related diseases, to cognitive dysfunction, to diseases such as Alzheimer's? What are the changes that occur in the brain but throughout the body? That's really what we're trying to understand. And this is important because a tsunami is approaching us, a tsunami of Alzheimer's disease. That, just to throw out a few numbers here, 32% of people at the age of 85 have this disease. And the disease is thought to start actually 1 or 2 decades before you get clinical symptoms, suggesting there is way, way more people who have this disease. It's a disease of the old. 81% of people who have the disease are actually older than 75 years of age. And you might think, well, you know, I'm not going to be 75 for a long time, but people are getting older across the globe. And the fastest growing segment of the population across the globe is people above age 65. So this is a problem that will face every nation on this planet, and we really have to understand the basis of this disease and how we can potentially prevent it. And the projection is by 2030, 75 million people will have the disease. So this is sort of all scary news, right? But I'm trying to show you that to just make you aware how big of a problem this is going to be. So wouldn't it be great if we had what is depicted here in this painting from Lucas Cranach the Elder, a fountain of youth. You probably have seen similar paintings and pictures. Every culture actually has some idea of a magic potion or a magic water that you can drink and it makes you young and rejuvenated again, such as these individuals here who enter the fountain and take a bath in it, and they emerge young and rejuvenated on the other side. So what I'm going to tell you, and this is really the basis of the research in my lab over the past 15 years, is that in mice, maybe there is such a fountain of youth. And maybe it even exists in humans. And maybe it has just dried out and we have to try to reactivate it, to regenerate and rejuvenate old tissues. And this all started in more recent time by an experiment that is somewhat creepy, that uses 2 mice of different ages, and they're sutured together at their flank. So what the scientist here, Tom Rando, who's a good friend of mine and has actually his lab next to mine, what he did is he made a cut in the flank of a young mouse and an old mouse, sutured their skin together, and after a few days, blood vessels grow into that wound, and because these mice are genetically identical, the tissue is not rejected, but the blood vessels actually fuse. And so now you have an exchange of blood from the young animal into the old, and from the old into the young. You still have the 2 hearts pumping against each other. So it's a very slow trickle of blood that goes from one animal to the other. But why on earth did he do this experiment? So Tom is interested in aging of muscle, and specifically of the stem cells in the muscle. When you're young and you exert your muscle very heavily, bodybuilding, your muscle gets injured, that activates the stem cells and you make new muscle tissue. But when you're old, these stem cells for some reason don't get the signals, and they make actually no new muscle tissue, they make scar tissue. And Tom asked a very simple question: is this because something inside the stem cell is missing or defective, or is it something from the outside? And with this model, he could address the exact question. Because here you have now an old mouse that has defective muscle repair, defective in making new muscle tissue, that is exposed to young factors. And what he found is that this old muscle regenerated and grew like young muscle tissue. When he looked at tissue from this muscle, it was indistinguishable more or less from the young muscle tissue. Suggesting, indeed, there are factors in the blood circulation that are carried from this young mouse into the old mouse and help the muscle stem cell regenerate and rejuvenate. He also observed similar effects in the liver, and yet some indication that in the brain there's also a growing number of cells that we call stem cells, that these stem cells were reactivated. But Tom didn't really study the brain, and that's how our collaboration started, and we decided to explore whether the same applies to the brain. And indeed, over the past 10 years or so, a number of labs have shown that this regenerated capacity of young blood applies to muscle, to the liver, pancreas, the heart, bone, the vasculature, and several groups now, including ours, show effects on the brain. An important discovery that my lab made, and specifically a graduate student who first did these experiments, Saul Vileda, is that you actually don't need to suture the mice together. You can simply collect blood from a young mouse and put it repeatedly into the old, or vice versa. And this is shown here. So what we did is we collected blood from young mice, and we found you don't actually need the cells in the blood. You just need the liquid part, which is called plasma. And you can inject this into the old mice, and what you see is increased stem cell activity in these old brains. You see more activity between neurons that we call synaptic plasticity. You see more electrical activity, less inflammation, and most importantly, improvement in memory function. So we have sort of a test that is similar to finding your car in a parking lot for mice, where they have to find a hole where they can escape on a platform. And there's actually a TED movie where you can see this experiment. What's also very interesting is the opposite is true as well. If you take plasma, the liquid fraction from blood, from old mice, and you infuse it repeatedly into young mice, then you reduce stem cell activity, you reduce the communication between neurons, you cause more inflammation, and you cause also more inflammation of the blood vessels, suggesting that there are bad factors in old blood, good factors in young blood. So that really triggered a whole number of questions in our laboratory. What are these factors? Can we identify them? Can we find the key factors that are involved in making these old mice younger or the young mice older? Where do the factors come from? What are the cells in the body that produce these factors? And why are they not producing them anymore when you're old? Which cells are the target of these factors in the brain, for example? Does it affect every cell or different cell types? How do the factors reach the brain? You may have heard about the blood-brain barrier, and I'll talk much more about this in the rest of my talk. So how do these factors actually get to the brain? And then that prompted us to more general questions about aging. Within a body, we know that every tissue gets older, but do they age at the same time? Do they age at the same speed? Are there some tissues that age faster? Are there cell types in a given tissue that age faster or slower? And if that's the case, let's say a liver cell ages faster than the rest of your body, can that cell actually spread aging? So can that cell produce a factor that then almost infects other cells to age? Such as we showed when we inject old plasma into young mice, we make these mice older. So what are these factors? So just first, a few experiments that we did to understand what are the factors. To do this, we look again at the blood. We look at blood from young people and old people. We've also done this in mice, but we really want to understand human aging. And we asked the simple question whether we can study aging by looking at the composition of blood. Does the blood change when you get older? And to do this, over the decades, people have developed more and more sophisticated tools, and this is probably one of the most sophisticated tools that we didn't develop in our lab, but that we were able to harness for the study, and this was done by a fellow in the lab, Benoît Le Hallier. And what we use here are, instead of antibodies that are frequently used to measure proteins, we use so-called aptamers. These are modified DNA molecules that specifically capture individual proteins. And here you see an example of one of these oligonucleotides binding the protein IL-6. So there's a company called SomaLogic that developed now close to 5,000 probes to measure 5,000 different proteins in your blood. And we have been able here in this study to measure the protein composition in over 4,000 people from young to old, and we measured over 3,000 proteins to really see how does the composition of these proteins change as you get older. And this is not a painting from Roy Lichtenstein, but this is how your blood composition changes with age. And what we show here is in colors, we show in blue low levels of a protein and in yellow high levels of a protein. And just to put the labels back on here, so these are young people, and here you have 4,000 individuals, and for each one of these individuals we report 3,000 different protein measurements. And so with the colors we show the levels of these proteins. What you can see is that there are a lot of proteins that are expressed at low levels, and when you get older, they increase. They tend to be in inflammatory or injury pathways, but there are also proteins that are high when you're young and they go down. But very importantly, what you also see is this is not a linear process. So for a lot of people, aging is sort of a linear process. You're young and then you get slowly older. But what we're showing you here is that's not true. If you look at the composition of the blood, you age sort of in a noisy way. Actually, it's not entirely noisy. So what we can do is we can take the first derivative of this, but before I go there, just if we look at the individual proteins and compare, for example, men versus women, we see huge numbers of proteins that are either positively or negatively correlated with sex. Roughly half of the proteins are different between men and women. But what we also see, as you saw before, is that large numbers of proteins, roughly again, half the proteins change from young people to old people, suggesting that your body lives in a very different environment when it comes to aging. So that's why it's maybe not surprising if you take the blood from a young organism into an old, or the other way around, you have profound effects on the organism. So what you can do with this is you can sort of play with the data a little bit, and we can make essentially a clock of aging. So to do this, we can take 2/3 of these individuals we measured, and we learn which proteins change in these 3,000 people, and then we predict the age of the remaining individuals. It's called a discovery set and a validation set. And what we can show then is that with the protein concentration in your blood, we can essentially tell you how old you are. And this is shown here. So here is the actual age that a person lived, and then based on the composition of the proteins in the blood, we predict how old a person is. If we would predict perfectly the actual age, which is called chronological age, then everybody would be on this red line, but we of course know that not everybody ages at the same speed. We think we can guess this from their facial features, but there's of course internally you see different organs age at different speeds. So for example, this individual here is 50, but we predict that person to be 35 or so. And this individual here is around 60, but we predict it to be 80. So here we can potentially predict the biological age, the really relevant age of the body of an individual, and we call that the delta age. And we can in fact show that this age, the difference between the actual age and the predicted age correlates well with function. So people who tend to be below this curve have better cognitive function, they have better grip strength and other features that we can measure, and people above it, they do worse, suggesting that what we predict here has something to do with function. And the idea is in the future that we will be able to extract individual proteins and figure out how they relate to the aging of an individual, to the aging of individual tissues potentially even. And so here, if we take the first derivative and we find out how many proteins change at each given age, from younger people to older people, what we find, if we then sum up the number of proteins that change at each given age, that if you're around 35 years of age, there is this large number of proteins that are different in younger people compared to older people, suggesting that there is a major inflection point of aging happening at a very early age that has not been previously appreciated. Most people, when they study aging, they really look at this peak here around 80, where massive changes are taking place. And then there's another little hump here where we can actually show that these peaks correlate with age-related diseases. A lot of proteins that change here are involved in neurodegenerative diseases. Proteins that change here are associated with heart disease, but these proteins here, we don't know why they change. And so we want to understand what is going on at this early age. Is this potentially a predictor of how you will age in the future? And what are the proteins that are here? And we can make lists of these proteins that change in each of these waves of aging. What are they? And in what biological processes are they involved? Can we learn something about the aging process with these? So the other aspect I want to discuss with you for the rest of the talk is how do these factors actually reach the brain? So I showed you massive changes with aging, and I showed you in work that was mostly published that we can take young blood and we can regenerate and rejuvenate the brain. We can take old blood into a young mouse and we can have the opposite effect. And actually should say also that we started a company that is translating this into humans and is now treating patients with Alzheimer's disease with plasma from young individuals to see whether this can be translated to a clinical setting. But the question remains, how do these factors that are in your blood, how do they actually get to the brain? How do they communicate with the brain? And there's sort of a dogma almost in the field that says stuff that is outside your brain cannot get into the brain because there's a blood-brain barrier. And this picture of a barrier that opens and closes, I think, is one of the worst things that we have in neuroscience because it's clearly not a barrier. But it has prevented a lot of people from studying this interaction between the periphery and the brain. And so when you look at a blood vessel here, so this is a cross-section of a blood vessel, there are other cells around it called glial cells, astrocytes, microglial cells, and here you would have these neurons. And what we were specifically interested in, and this was a graduate student who was in the lab, actually a bioengineering student, incredibly talented, wanted to understand how proteins get transported from the blood into the brain. And you can imagine different ways how something from the blood could get in there. It's possible that these cells here, these endothelial cells, are not completely connected with each other, and that under certain conditions, there's openings where factors could non-specifically just get in. There could be possibilities where you activate the cells in the blood vessels, and these cells then communicate with the brain. Or you can have active transport mechanisms into the brain. And so we wanted to understand how this transport happens, which ones are important, and how it changes with age. So again, if we look at this interaction between the blood, so this is a blood vessel, here would be the blood. These are the endothelial cells, so these are the vascular cells that form the blood vessel. And then we have other cell types that are important to maintain and form this specialized vasculature that we have in the brain that is very different from the vasculature in the rest of the body. The way people have studied this in principle is as follows. So we know that there are two major types of transport. One is called receptor-mediated transport, where you have a specific receptor that binds a protein such as transferrin or insulin, and then forms a clathrin-coated pit that transports molecules across the cell and releases into the brain tissue. A second mechanism is non-specific. It's called adsorptive transcytosis, and is based on charge interaction with the cell surface of the endothelium. It forms a different type of vesicle that also can transport things in. So this is specific, this is non-specific. The brain has evolved a mechanism that reduces this non-specific transport by about 90%. So you have very little non-specific transport, and that's why we have this, what people call the barrier. So the way this barrier has been studied in the past is mostly by artificial molecules or proteins. One is a protein, an enzyme called horseradish peroxidase that has been used to study whether this protein can get into the brain or not, because you can easily detect it and you can detect its enzymatic activity. Another protein is called Evans blue. That's a small chemical that binds to albumin, the main protein in the blood. And so you can measure how much albumin gets into the brain. And if you inject this, both of these do not enter the brain in a normal condition. And then the third one are these different types of sugars that can be very small to very large. And again, these were used to study the blood-brain barrier and come up with this concept that almost everything is kept out of the brain. Of course, these are not natural proteins. They're not what the brain is exposed to. These are artificial substances. And so what we argued is, if we really want to understand what this barrier transports, which of the proteins in the blood can actually get into the brain, we have to study the proteins that are in the blood, right? We can't study some artificial molecules that are not actually in the blood. And so if you just look at the composition of the blood, most of the proteins are made up of very few proteins. Albumin is about half the proteins, and then you have antibodies and a few other proteins that are highly abundant, but most of the proteins are present at very, very low concentrations. So what we decided to do is to label all these proteins rather than to look at one individual protein at a time or even look at foreign molecules to test this barrier. We decided to label all the proteins at the same time. And we used different methodologies. We used radiolabeling, we used highly fluorescent tags, and we used a biotin molecule to then recover these proteins from the brain. And so we used different types of technology such as radio tracing or next generation sequencing to try to identify what kind of proteins make it into the brain. And so if you just inject labeled plasma into the circulation of a mouse, what you end up, and then you take out the brain and you make the brain transparent to see where the plasma proteins are. First of all, what you see is there's lots and lots of plasma proteins in the brain. Now, most of what we see here is in the ventricles, so the hollow spaces inside our brain, but then you also see a lot of protein that is in different other areas. And so one of the classic technologies that has been used is autoradiography, where you label proteins with a radio tag so that it's radioactive, and then you can make brain sections and you can actually image where the radioactivity has gone. And if we use an antibody, which is traditionally used in these studies, we can see what many other people have reported. You see uptake into the ventricles, and then some here in the hypothalamus, where this barrier is thought to be not as tight and relatively open. If you inject now plasma proteins, what do you think you will see? More or less? You actually see much more. So this is plasma proteins labeled with the same technology. We actually take out the antibodies first, and we also take out the albumin. So the most abundant proteins we deplete. So we label here only the lesser abundant proteins. And when you do that, you see there is more uptake actually than with an antibody. And there is also uptake into many other brain regions that we can detect. And if you quantify that, there's a several-fold increase in plasma that is being taken up into the brain compared to a classical antibody as a control. And that's the case in different brain regions. If you look at the brain, so now we look at the slice of a brain from a mouse that was injected with labeled plasma, and this is a fluorescent tag now. What you can see is this beautiful labeling of the whole vascular structure. And this is a brain that was washed and perfused over and over and over. So it's not what is in the hollow of the blood vessel, but it's what either made it into the cells or outside of the cells. And we see again, a lot of proteins are in the ventricular spaces, but there's, and also in the hypothalamus, but you also see sort of foggy staining in other areas. If we zoom in further at individual blood vessels and trees of blood vessels, you see this beautiful, gorgeous punctate staining. So these are all little vesicles, little bubbles that are filled with plasma proteins, suggesting there is massive uptake of plasma proteins into the vasculature. And it reminds me a little bit of the Christmas decorations that you see in many places here at Times Square. But what's really exciting is that we don't just see this labeling in the vasculature, but we see areas, for example, in the hippocampus, this key center of learning and memory, where actually neurons take up plasma proteins. So here we label, again in white, is plasma proteins. This could be any protein that is in the plasma. And in blue you see the nuclear marker. We see also, if we label here the blood vessels, we see these beautiful large neurons, probably excitatory neurons, that are full with plasma proteins. And here another image in the hippocampus again, labeling of blood vessels as well as of neurons. So we wanted to confirm that this is really true and we're not just chasing an artifact here. And one of the proteins that is very well known to be transported actively into the brain because it's required there is this protein called transferrin. And transferrin transports iron into the brain. So if it carries iron, it's called holotransferrin. It binds to the transferrin receptor. It forms these clathrin-coated pits that then shuttle transferrin into the brain and deliver the transferrin with the iron to brain cells, including neurons. So what we did in these mice is we injected the labeled plasma in green, so all proteins labeled in green that we inject. And then we used an antibody specifically recognizing transferrin, and we labeled this antibody in red. And what you see is, as people have shown before, there is transferrin in the brain, and so you get all this red staining. But then you also get green from plasma proteins. And where they overlap, we demonstrate that transferrin comes from outside as well as locally. So showing that some of the transferrin that we can detect here is actually coming from the outside, from what we injected, proving that our method of detecting proteins in the brain is indeed accurate, and we can show this also with other proteins. We see also plasma protein deposits in cells other than neurons, for example here in microglial cells. Very few microglia take up these proteins that we're now trying to characterize, so we can isolate these microglia now and we can figure out what are these cells and what are the proteins inside these cells. And we think they might have a role in aging and neurodegeneration. We used also another method where we labeled the proteins with yet a different method where we incorporate amino acids into the whole proteome of one mouse, and then we transfer this labeled proteome, so this is labeling for the aficionados, with AHA, acetohomoalanine, so this is an analog of methionine that now gets incorporated into the proteome of this mouse here. Then we take the blood of this mouse, which has now proteins labeled, tagged, we inject it in another mouse, and then again use these detection methods. And again with this method as well, we can show uptake of plasma proteins into blood vessels, as well into neurons and other cells. Showing now with 4 different technologies that this is real. There's massive uptake of proteins into the brain. And we have data that I don't have time to show you. We're now building a catalog to show what are the proteins that accumulate in the brain and what proteins accumulate in different cell types. Now, one of the questions you might ask, is this a regular and even uptake into the brain? Or are there differences, and what are the rules that lead to uptake of proteins into the brain? And to address that question, we used a new technology that is called single-cell RNA sequencing. And you can actually see here that not all these blood vessels, it's not an even distribution of these green dots. There are some cells that seem to have less uptake and others have more uptake. So what we can do with new technologies now is we can take the brain of a mouse that was injected with labeled plasma. We dissociate the cells into individual cells, and now every cell is labeled by how much protein it took up. And we can sort them so that we can then correlate the relative level of plasma uptake. So a cell may take up a lot of proteins, and then we measure the genes that are expressed in each individual cells. So now we compare what genes are expressed in a cell that takes up a lot of proteins and what genes are present in a cell that takes up no proteins. And when you do that, you get this beautiful Gaussian curve where you have most genes have nothing to do with uptake of proteins, as you would expect. So most genes that are expressed in these cells in the blood vessels have nothing to do with uptake. But you see a small tail here of genes that when they're expressed, they increase, they highly correlate with uptake of proteins. And what's very exciting is that one of the top hits is this transferrin receptor that I showed you before. So this is a protein that is involved in uptake of proteins into the brain, and we can show here if it's highly expressed in a cell, they take up more protein. And we found a few other ones. This one in particular here, SLC3A2, is an interesting one. There's a whole effort in pharma industry, in biotechnology industry, to use these transporters to bring drugs into the brain. Because of this concept of the barrier that things do not go into the brain, people use the transferrin receptor, or most recently Denali Pharmaceuticals, they used this transporter here, which they discovered in a gargantuan effort, and it came up as one of our top hits as well, to shuttle proteins into the brain, especially antibodies. They want to get antibodies against amyloid, for example, into the brain, and they use these to shuttle them in. So we discover here these positive transporters that bring things into the brain. But what we also discover for the first time that has not been reported before is what's on the left-hand side here. So here we call these anti-correlates. So these are genes, when they're highly expressed, there is no uptake of proteins into the vasculature. So these genes are highly expressed in the cells that don't take up proteins. So what if these are actually inhibitors of protein uptake? And if you wanted to get more proteins into the brain, you would target these as proteins. And I'll talk in a moment about one of them that we're pursuing here. When we look in what kind of biological categories these factors are expressed, these genes, we find the correlates are mostly in specific receptor-mediated transport, vesicle-mediated transport, so all the specific transport that I showed you at the beginning, where you have a receptor that recognizes something and then transports it in. These are mostly on this side. What's very exciting and interesting, curious, is that these inhibitors here, look at this, ossification, calcification, something with bone formation here. And it's interesting that with aging, you get actually calcification of the vasculature, you get bone formation in some types of diseases, and we can actually show this now in very old mice. So there's something related to a pathway that has not really been linked at all to vascular aging that is present in these cells that do not take up proteins. So we're trying to understand that. So now you may wonder, this is all good, this is in young mice that I showed you, but how does that change with age, and how does that relate at all to neurodegeneration and Alzheimer's disease? Why should I care? What I showed you so far changes dramatically with age, and this has not really been appreciated before. So let's look again at the radioactivity labeling. If we label again an antibody, the classical tool that people have used to study uptake into the protein, you see in a young mouse, I showed you a similar picture before, this is another mouse, you see this uptake into the ventricles, and with age you see more uptake. Everybody has described this is well known, people call it leakiness of this barrier. And you see there's a little bit more uptake here in mice. But what do you think happens to the plasma proteins? I showed you with young, there's about a 4 or 5-fold relative increase in protein uptake in young. But what you find in the old is actually the opposite happens. So you have a lot of uptake in young, but much less in old, and we have actually different time points in between. You see there's roughly a 50% reduction in uptake of these plasma proteins into the brain. And this is again without antibodies or albumin present in there. We can also show this at the transcriptional level when we look specifically at these genes that are involved in the specific transport, receptor-mediated transport receptors or clathrin and these adapters that are necessary to form the vesicles. They decrease with age. There are lower levels of these genes in most of these mice. On the other hand, genes that are involved in the non-specific transport go up. Proteins such as caveolin as well as other adapters go up with age. And what we can show is that many of the genes that are down with age are again involved in specific uptake of proteins into the brain. So these cells that take up a lot have high levels of these proteins, and they go down with age, suggesting again there is a switch from specific to non-specific transport. And I just want to illustrate this here with a few specific examples. So again, we have this specific uptake into the brain, specific receptor-mediated transcytosis that uses this protein called clathrin. This is an indicator for us for this type of transport. And when we look at vesicles that have this protein, you see lots of them in young blood vessels, but much fewer in old blood vessels, roughly a 50% decrease. On the other hand, the non-specific adsorptive transcytosis uses a different protein to form these vesicles, uses a protein called caveolin, and that is present at low levels in young blood vessels, but increases several-fold in old blood vessels. Again, supporting this concept that with aging, there's a massive switch from a specific transport of proteins into the brain to a non-specific one. And lastly, if we look at individual proteins, such as transferrin, which I talked about before, which is specifically transported, because there is a decrease in the specific transport, we have actually less transferrin getting in. So using this shuttle to get drugs into the brain in old people is not a good idea. And a lot of companies are pursuing this now, and I think it's not the best way, the best shuttle at this point. On the other hand, as others have reported, non-specific transport, albumin is a substrate for the non-specific, goes up with age. And we can show that also with another protein here, leptin, which is massively transported into the brain. That too goes down with aging. Specific transport goes down. And another non-specific transported protein, HRP, which is this tool that was used, goes up with aging. So can we manipulate, in the last few minutes, can we manipulate this and can we restore usefulness in the vasculature? Can we restore that transport to youthful levels? That's what we're trying to do, because we believe that this abnormal transport from young to old, that this dysfunction in transport may be in part responsible for why the brain experiences all these detrimental symptoms. So one of the proteins that we picked here is this negative regulator of transport. So this is an inhibitor of transport. And what we're going to do now is we will inhibit the inhibitor. To basically, this is like a brake, and what we're going to do is we take the brake away. And to convince you that this is a good target, what we found is that in young blood vessels, this protein is barely expressed. It's very low level expression. But in old mice, and this is roughly an 80-year-old person compared to humans, in this old mouse, there is a lot of protein expressed. And so if this is an inhibitor of transport, we want to block this. And the way we do this is we use a drug that is actually being described before that targets specifically this enzyme, which is present in the rest of the body, but not usually in the brain vasculature. Only with aging it gets expressed there. So we use this drug and treated mice with it, and we can indeed show that there is more uptake with the inhibitor. There's more plasma that gets into the brain, and there is also more transferrin that is being transported in again. So restoring some of the defects that we have observed with the aging vasculature can be restored with this drug here. That's a relatively crude drug. It's actually one of the World Health Organization's 10 essential drugs that was used for worm infections, that we try to repurpose here and actually make better analogs of this drug to target this protein to restore transport in the vasculature to youthful levels. So what I showed you is that in young blood vessels, there is an extensive communication of these thousands of proteins that are present in the blood with the vasculature. Many of them are transported in. They end up in neurons, they end up in other cells. We don't know yet what they're doing. But that transport shifts dramatically as mice get older, and we would expect to see similar things in human brains. And along with that, actually other aspects of the vasculature degenerate. We see also a loss of these support cells that the vasculature has, and we don't know if this is related to the transport. And then we see this accumulation of calcification and formation of structures that you don't usually see in young brains. So we think this is a mechanism for age-associated leakiness that people have previously reported because you have more non-specific uptake of proteins into the brain, or potentially even other particles. We talked earlier about viruses potentially getting it transported into the brain through this pathway. It could explain the increase in toxic plasma proteins with age, such as antibodies. It opens the question about the physiological relevance of cellular plasma protein uptake. So again, what are these proteins all doing? How do they get into the brain? What are they doing there? Are they necessary? Are they fulfilling essential function? And if they change with age, is that detrimental? And then lastly, this would predict that reduced uptake of this specific proteins could be targeted, and that could lead to the development of new shuttles. And with shuttles, I mean ways to bring drugs into the brain, drugs that are already available but don't have a good transport into the brain. We could improve that transport using this knowledge. Or we could identify new types of shuttles that could be used to hijack sort of, or piggyback on rather, to get drugs into the brain for the treatment of brain neurological diseases and neurodegenerative diseases. And to just put this in perspective, changes in the vasculature are incredibly prominent in the aged brain. This is a study, it's a very famous study, a religious order study from the Russian Memory Center, where they looked at over 1,000 brains from people who passed away from all kinds of different diseases, some no overt disease, they just died of old age, which still happens. And if you look at individuals who have no dementia, you see roughly 2/3 of them have vascular pathology. They have changes in the composition of their blood vessels. And this is just looking structurally mostly. This is not looking at the molecules that I showed you, but I think a lot of it has to do with this. So supporting what we see in the mice, that the vasculature changes dramatically just with normal aging. If you look in Alzheimer's disease, this goes up to almost 80%, so even more vascular damage, even more vascular changes. And this is something that the field has largely ignored because it's hard to understand, it's hard to grapple with. Most of the field has focused on these individual 2 lesions that we see, the amyloid plaques and the tangles. And the drugs so far have not been working there. So I propose that we should focus much more on this direct interface between the body and the brain, which are the vasculature. And I'm of course not the first to say that. This is a wonderful review by Beca Slokovic that talks of all these different aspects of changes that occur with Alzheimer's disease. So he has studied this mostly in a disease setting, but what we're showing here now is that this happens with just normal aging without having a disease, and we show for the first time what the molecular basis is of many of these changes. And with that, I would like to thank a lot of collaborators on many different aspects of this project. A wonderful lab of very talented students and fellows, and then funding from these sources here. Thank you. Questions?
Young versus old animals. Have you had a chance to look at young versus old blood vessels in the same animal? In other words, like what would you see if you had an older animal and had angiogenesis or something, if you took one of those vessels?
So you mean from the same animal or just an older animal? So what we looked at blood vessels from old animals. There is almost no angiogenesis anymore in old blood vessels. In fact, you have sort of a loss of the vasculature. You have actually less blood vessels. Sometimes you find only the matrix left, or you find these ghost streaks. And the cells that form the vasculature, they're actually, like the pericytes, they're also lost with aging.
Under certain circumstances, you can have these vessels, like moe.
Yes, in specific types of diseases you can have angiogenesis and you can have excessive blood vessel formation. Yeah. And also I think after stroke you get some revascularization, but with what seems with normal aging we see very rarely any type of proliferative cell in the vasculature.
In your experiments, what is the period of exposure necessary to affect clinical change?
Yeah, so in the mice, initially, just because we had nothing to guide ourselves on, we decided, so the limit of what you can inject in one injection is about 5% of the total blood volume in a mouse. And so we decided to do this every 3 days for 3 weeks. And that's where we saw the changes that we reported. Other people have then tried it, including our lab, but mostly the company we started, they have then tried all kinds of different paradigms, and it seems one of the most effective is if you give daily infusions, either once or twice for 5 or 7 days. And with that paradigm, what they see is sort of multiple events. You see after every injection, a few hours later or an hour later, you see already activation of neurons. You see CFOS activation throughout the brain. And that may be an immediate sort of neurovascular coupling response or something. We don't know what that is. And then you see after a few hours, days, you see changes at the cellular level. You see reduction of inflammation. And then you see long-term effects on neurogenesis that last for several months. Even after a 1-week treatment, you see several months later, you still have more stem cell activity. So for the clinical studies that are ongoing or have been conducted already, what they decided to do is to bring patients in, give them 5 infusions once a day for a week, and then 3 months later they come back, they get tested, they get another round of infusions, and then after 6 months total is the conclusion of the study.
Would you imagine that there's a possibility with a single exposure that there could be some kind of clinical difference in an individual?
Yeah, I think there could be an acute effect. As I said, you get an immediate activation of neurons and probably also glial cells if you look at the transcriptional level, but probably not a long-lasting effect. So I would not be worried if you need a blood transfusion that would have an immediate effect. Also, usually if you get blood transfusion in an emergency setting, you get donations from different people, and some may be younger, some may be older, but you wouldn't get just from old individuals, right, blood. Plus the blood products that come from these major blood product companies that process tens of thousands of samples every year, or hundreds of thousands of samples, they have an average age of about 35 years. So if you get plasma from one of these companies, then the average age is still very young.
Have you been able to correlate inflammation, autoimmune disease conditions, which is a basic inflammation that you get when you get older, various diseases, with the effects on the cells in the brain?
Yeah, that's a very interesting question. So we see in general with age, there is an increased activation of immune pathways, and some of them might actually be autoimmune in nature, that you get sort of, especially antibodies that may recognize your own body. And I would think that they might be detrimental for the brain. But there's definitely a massive activation of immune and inflammatory pathways throughout the body with aging. This is one of the key signatures that you see in almost every tissue. We don't know why.
Assuming some of us are aging, what actions can we take today and tomorrow?
Yeah, we're all aging. You know, I think there are, from animal studies, there's sort of interventional studies, and that show that caloric restriction, for example, or intermittent fasting is sort of a buzzword now, where animals, and a lot of people practice this now, sort of mimic the natural setting, how we used to get food. We used to have food, on good days we had a lot of food, and then we had periods with no food. And most animals have that. And what people find is that if you have an extended fasting period, not just the 9 hours that we sleep, and we still use the term break fast, right, in the morning. If you extend that to 24 hours, then that's beneficial for many organisms. Others are, of course, exercise. Lots of studies show that exercise is beneficial and reduces inflammation in many different tissues, including the brain. Healthy diet. But there is no magic potion.
It was a great talk.
Thank you.
One question, to bring the two halves of it together, if you transfuse plasma into, say, older animals, do you reverse the transport bias back towards receptor-mediated transport?
Yeah, so that's a very interesting question. We haven't done this experiment yet. So right now, when we do these transport studies, we infuse the plasma, and then just 4 hours later, we look in the brain, so that we don't have a rejuvenating or an age-promoting effect. And of course, young into young is not a problem. But also in the aged brain, we usually inject young plasma, but we look only 4 hours later. And so in that period, we don't think we change the composition of the blood-brain barrier. But I think you know, what we want to do is to do exactly this experiment, take aged plasma, give it into young, and see how we change these parameters and which ones we change, and then the other way around. We have done this experiment. There's actually a paper that will come out very soon, where we just look at the transcriptome of the endothelial cells. So if you measure the transcripts of young and old endothelial cells, you see massive changes in gene expression. If we give an old mouse young plasma, some of these transcripts go back to a younger age, and vice versa. If we give a young mouse aged plasma, then the transcriptional signatures become older. So it supports sort of this concept that aging is a plastic phenomenon. It's not like we used to think that it's just a one-way street, but you can indeed impact the aging process if you know how to do it, and it's still somewhat plastic.
In thinking about the dynamics of barrier function, there's interest, as I'm sure you know, about changes, variability over the course of a day.
Right.
Sleep and rest and so forth. So how did the change, first of all, have you investigated that as a function of age? Whether, and I don't know whether we're talking about the non-selective or the receptor-mediated, and the amount of change, that day-night, sleep-rest change, how does that compare with the aging change in terms of amount?
Yeah, we haven't done that yet. So we try to do the experiments at the same time of day, or also when we take down mice, we always try to do it sort of at the same time because of these well-known circadian changes. And I don't know what the magnitude of change would be, but I think there's probably a significant difference. We know from many other studies, cell types, glial cells, where you show these beautiful waves that you see, but also the composition of plasma changes from day to night, and that's something that is not clear what the impact of that is. And my guess is some of the proteins will be overlapping with the ones that change with age, and others will be different probably. it is interesting that with aging, so we have a separate study where we look globally at all major tissues in the mouse from very young to very old, 10 different time points. Among the top 20 genes that change across all tissues are circadian genes. There's a very strong circadian network that changes with age. And it supports what people have already suspected or know, but it seems to be in every tissue you find that signature. Yep.
Thank you for a wonderful talk. Have you correlated proteins in the blood with CSF proteins and correlated changes in CSF between young and old animals and humans?
Yeah, we've done some of that. And there's a fair number that get good correlations. Leptin, for example, is almost at the top, where you have very high correlation between levels in blood and CSF. And some of them are actively transported into the brain, either through the vasculature or potentially also through the choroid plexus, where they may have access. And we actually did studies where we injected at the same time labeled proteins into the cisterna magna or into the vein to look at these dynamics. But you see also massive changes in composition of the CSF with age. It's not as deep. We didn't have the same platform. We measured only a few hundred proteins, but there's clearly also about a third or so change.
Has anyone tried to transfer CSF from young animals to old and vice versa?
I have a crazy fellow who is doing that right now. And she sees very profound effects. Yeah, so she collects CSF from hundreds of mice and infuses it into the other age. Yeah.
So a quick question about another systemic factor that I'm curious to know whether these transporters are mechanosensitive and whether blood pressure modifies them and with the well-known risk factor of hypertension.
Yeah, I don't know, but it's a very interesting question, yeah. I mean, we see more and more these piezo receptors that show up in all kinds of different tissues. I don't remember seeing it, but it's something we should look at, yeah. I'm sure, so what we find, as you know, there is, obviously blood vessels are zonated from arteries to capillaries to veins, and there's a transcriptional program that allows you to identify cells based on zonation. And we find that most of the protein uptake is in the venous side, where you have the lowest blood pressure. And there's relatively less in arteries, and capillaries are sort of all over the place. Some take up a lot, some take up little, and that's what I showed you, where we can really regulate that transport. Mm-hmm.
Well, thank you all for coming to this event, and this was a fantastic, interesting, curious, wild idea, which I'm hopeful about the future for, and thank you so much for sharing.
Thank you.